in the vicinity of cells, it can potentially bind to the cell surface and enter the cell. And with that, there we go, that travels in. And once it's inside the cell, then the virus can uncoat and the DNA gets delivered to the nucleus. So you see the gene encoding the therapeutic protein. And next, we'll show that once that's in place, that gene can now be transcribed into RNA, and the RNA can be translated into protein. And that protein can be any type of protein, one that's located inside the cell, maybe across the cell membrane, or one that's secreted. The main issue in gene therapy is trying to find the right type of virus and the right gene for each disease. Next slide, please. So in terms of what so as we mentioned, you choose your virus selection based on what cell types you want to deliver the gene to. You have to choose what gene to deliver, and next. And then you also have to decide where in the body to deliver the virus. So, for example, if you want to deliver a gene to the brain, you might inject it in a different place than you would if you want to deliver to the muscle. Slide, please. So one quick example of this is an example I mentioned before. It's Hemophilia B. This is caused by mutations in the gene encoding factor IX, which is part of the clotting cascade. So in patients with hemophilia B, they have mutations in this gene. It results in a lowered ability of their blood to clot. They will bleed heavily after injury, and depending on how severe their disease is, they can also get spontaneous bleeds, which can be quite damaging and even dangerous. So treatment that's being tested in the clinic now by multiple groups is an intravenous injection of a particular type of virus called AAV for short, which carries a normal copy of the factor IX gene. And so this virus can be delivered through a vein, and when delivered, most of the virus goes to the liver. And once that is delivered to the liver then starts synthesizing and secreting factor IX, which can then circulate where throughout the circulation, which is where it's needed. So it's kind of turning the liver into a factory to make normal factor IX and to reduce the symptoms of that disease. Please. A brief history of milestones in gene therapy. In 1989, a first clinical trial was done, which actually used a type of virus called a retrovirus. It introduced a marker gene. It wasn't a therapeutic trial. And next shows that a year later, the first gene therapy trial was used to treat disease. So again, the same type of virus, retrovirus, was used to introduce a therapeutic gene in cells of patients with a form of severe combined immunodeficiency. Next, please. Then there were some setbacks in the field, so more trials began ramping up. In 1999, a gene therapy clinical trial for ornithine transcarbamylase deficiency using an adenovirus vector resulted in a severe immune response that caused multi-organ system failure and the death of a patient. Another setback happened several years later on the next, which was a clinical trial for another form of severe combined immunodeficiency. This one using a retroviral vector resulted in leukemia in several patients. But most of those patients were successfully treated for the leukemia. But the other thing about that particular clinical trial is all the surviving patients were successfully treated by the gene therapy. So although it was a setback, there was also a big clinical benefit to those patients. And the next show, the next sort of thing that happened where vectors became improved, meaning that people were able to deliver genes more efficiently to patients. And as I mentioned before, these AAV vectors, they're adeno-associated virus vectors developed with enhanced levels of gene transfer. The next also shows that in those leukemia patients, the type of vector being used was replaced. So a different version of retroviruses called lentiviruses were developed, and clinically all the patients treated with these have not had any leukemias to date. So that's been, and have been successfully treated. So that's been another good advantage. And then on the next, more recently, there have been some approvals. So in 2003, China approved agendicin, which is an adenovirus for tumors with specific mutations in the p53 gene. So this delivers a normal copy of that gene. And on the next slide, the next shows that in 2012 in Europe, a compound called Glybera, which is again an AAV vector, but it's for an inherited disease called lipoprotein lipase deficiency, and that was approved in 2012, which was very exciting for the field. And so I think, okay, so now as of 2015, over 2,200 gene therapy clinical trials have been initiated worldwide, and of those, that has included about 100 late phase or registrational trials, just to show that the field is moving further towards getting real therapeutics in this area onto the market. And with that, I will turn it back to Danny for the next one.
Sharon, there's a certain elegance to gene therapy, but in practice, it's not as easy as it might seem. What are the biggest obstacles therapeutic developers face in getting gene therapy to work?
A couple of things that are definitely big ones. One is the manufacturing piece. So making the gene therapy itself, these vectors, is a very complex procedure, can be very difficult to scale up. The product itself is more complex than, say, a small molecule, which is typically in a pill form. So manufacturing has been an ongoing challenge, but one that's being met and developed as we speak. And then the second piece of it is really understanding how the data that we get from animal models, which is usually where most of the preclinical work is done, is in animal models of disease, and understanding how the data we get from there really translates into human efficacy. That's always an obstacle because you can't predict everything from the preclinical work you do. And so this includes things like effects of the human immune system, what the levels of expression are when you move this from a smaller animal into a human, and that will simply take some time in terms of getting additional data from the clinic.
You talked about Hemophilia B. Can you offer some other examples of rare disease therapies in development today where we've seen promise that are advancing through clinical development?
Yeah, so right, the Hemophilia B clinical trials are still currently in the earlier phases, meaning phase 1 or 2 out of the typical 3 phases of development. Other things that are advancing further through the clinic is Bluebird Bio has an lentivirus in development for another rare disease, cerebral adrenoleukodystrophy, and that's currently in pivotal clinical trial, so that's a phase 2/3 clinical trial, and they're starting to release some data around that, so that's very exciting. And also Spark Therapeutics is developing another therapeutic for inherited eye disease called Leber congenital amaurosis, and that they've completed now a phase 3 clinical trial, and they're planning to apply for registration within the next year.
What we know about the durability of gene therapies today, are these one-time curative treatments, or will they need to be readministered over time?
Yeah, that's a great question. So what we know so far is there's a lot of long-term data from the Hemophilia B patients. So the earliest patients treated have now been observed for over 5 years. And all of the patients in the group so far have had very, very stable levels of factor IX gene protein expression over the time they've been observed. So for some, it's over 5 years. So, so there is some long-term data. That is from a single treatment, a single injection with a therapeutic. So as of today, we don't know if those patients will need to be retreated, if it's not durable, and we don't really know the length of that expression. So far, it's been quite stable, but there is certainly the possibility that these maybe need to be readministered some years down the road.
I had a question from the audience. They're asking if the vectors are unique to the individual patients, but to what extent do these therapies have to be customized or be off the shelf?
So it depends on what the treatment is. So for something like the Hemophilia B treatments that are being done with AAV, that is an off-the-shelf treatment. so that every patient can be treated with the same product. For other diseases, such as the severe combined immunodeficiency, even the current clinical trial in adrenoleukodystrophy, that is a unique treatment for every patient because a patient's own bone marrow or stem cells is what is treated with the gene therapy vector and then reinfused into the patient. So that's different in that case. Go ahead.
We've had Abeona, as you mentioned, approved in Europe, and GSK recently had a positive recommendation from the EMA for its gene therapy stimulus for SCIDs. When do you expect to see an approved therapy in the United States, and any sense how regulators here view gene therapy?
So the thing that's closest to approval in the United States at this point is the therapeutic I'd mentioned from Spark. So they call it SPK RPE65, and it's for this inherited retinal degeneration that causes blindness called Leber's congenital amaurosis. And that, potentially could be improved within the next 1 to 2 years. I think that, interactions with the regulators in the U.S. are very positive. I think they're very interested in helping gene therapy get to patients. There's good expertise these days within the agency, so they give very thoughtful feedback. They're open to discussions, and they are, really working hard to balance the acute need for therapies with the need for keeping patients safe.
I want to remind our audience that they can instant message questions that they have for our panelists as we go, and we'll try to work them into our conversation as best we can or save them up for the end. But at this point, let's turn to Michelle Berg, Vice President of Patient Advocacy for Abeona Therapeutics. We're working to bring gene therapies for rare diseases to market. Previously, she worked for Aldevron, a contract manufacturing organization specializing in gene therapy, and she's also performed research in the Plant Sciences Department at North Dakota State University, where she did a BS in biotechnology. Michelle will provide a perspective on gene therapy from the point of view of a company seeking to develop and commercialize these therapies. Mel?
Thank you. Thanks also to Global Genes for hosting and assembling this webinar. So my next slide. Go up. So this is a forward-looking statement. Abeona is a publicly traded company on the NASDAQ exchange. So I'm asked how to pronounce the company's name, what it means, and where it came from. So see, I tried to spell out phonetically that it's pronounced Abeona. And this name selected by our CEO, a Roman goddess by the very same name. She's the protector of children as they begin their journey. And as you'll hear, the kids and families that we serve are on a significantly challenging journey. Next, please. Abeona was founded just over 3 years ago by a small team who licensed technology from Nationwide Children's Hospital, Columbus, Ohio. Doctors Doug McCarty and Hien Fu are the scientists there who worked well over a decade on Sanfilippo syndrome, now our front-running gene therapy programs for MPS 3A and 3B. Through support and help of about a dozen worldwide foundations, we raised about $6 million to manufacture the drug and bring ourselves to the place of being able to initiate clinical trials. You see represented here are what we consider to be our founding foundations, those contributing, as I mentioned, financially, but also through their belief in our approach using gene therapy and providing information and helpful feedback along the way. It's important to note that these groups range in size from single families all the way to national and international foundations. As you see, these come from Spain, Switzerland, Australia, Mexico, in addition to the U.S. And without them, we're the company that we are, and we wouldn't be able to do the things that we're doing today. Slide. So in May of last year, Abeona LLC completed a reverse merger with Plasmatech Biopharmaceuticals. Was a publicly traded company that was already specializing in rare diseases. So about a month after that, the company rebranded back to the Abeona name, kind of reconfirming our focus on rare diseases, particularly those that are impacting children. As it happens, just showing the picture here, my first day for the company was the day that we were able to come in and ring the closing bell at NASDAQ on June 22nd last year in celebration of this rebranding. And I have to say it was pretty much the best first day on the job that one could ever have. So here you see a snapshot of our current pipeline, or an overview of the progress of the therapies that we're developing. And so for the purposes of today's talk, I'll be focusing and sharing more about Sanfilippo syndrome types 3A and 3B approaches, as well as our program for juvenile Batten disease. Here on our pipeline, that ABO 102 for Sanfilippo A is our most progressed program to date. I'm very, very happy to share that we received allowance for our phase 1/2 IND for this program in February of this year. So it's a major accomplishment, not only for our team here at Abeona, but more so for the collective group that supported us since early 2013 at the foundation. So the three programs that I'm going to focus on, these are initially been set and focused on lysosomal storage diseases, of which there are over 60 types. These are recessively inherited diseases that are caused by a defect in a single gene and result in a missing or malfunctioning enzyme. It translates to basically a buildup of waste in the lysosomes, which is part of the cell that would otherwise be responsible for removing such waste. So as you can see in the graphic here on the right side of the image, there's a buildup within these lysosomes, which is shown as the large white spots, and that's significantly different by comparison to the photo of the unaffected cell on the left. Okay, next slide. Sanfilippo syndrome, also called mucopolysaccharidosis, or in this case MPS 3, of which there are 4 different versions, A through D. Abeona is focused on the 2 most common forms, A and B. These are due respectively to missing or poorly functioning SGSH and NAGLU enzymes, which over time, again, allow for buildup of sugars in the kids' cells. It's really important to note that these sugars build up not only in the brain or central nervous system, but throughout the entire body and causing problems. So in the majority of cases, parents are presented with a seemingly healthy baby that will continue to hit milestones on target, if not early at times. And then sometime after about age 2, the parents start to see a slowing of development that eventually will turn to a regression and loss of function. So kids, they can demonstrate aggressive behavior. They have a very hard time with sleep, and eventually the decline in ability to speak, walk, and feed themselves is inevitable. And so it's particularly dating as a lysosomal storage disease because about 7% of these kids won't see their 18th birthday. So juvenile Batten disease is, again, also a lysosomal storage disease. Another name for it, juvenile neuronal ceroid lipofuscinosis. This can be traced back to an inherited mutation in the CLN3 gene. So in buildup of sugars, as with MPS 3A and 3B, this time it's an accumulation of lipopigments and proteins that, again, in the kids' cells. And nothing obviously wrong or apparent at birth. Kids oftentimes aren't diagnosed in this case until well after age 5, typically because of a combination of loss or vision problems and seizures. And like many in rare diseases, the road to a correct diagnosis is challenging, frustrating, and it certainly is much longer than it should be. Kids with juvenile Batten also will have troubles with sleep and behavior, and like MPS 3A and B, this is a disease that also impacts the entire body and over time will result in loss of everyday functions. In case we're very fortunate to be working with Dr. Tammy Killian out of the University of Nebraska Medical Center. She's demonstrated extraordinary results already in the animal mouse model, juvenile Batten disease, and her work reinforces what we've already seen in the research coming out of nationwide for the Sanfilippo programs. As mentioned, it's really important that we select the right virus for delivering the gene to the right place for the right disease. And so with that, Abeona has selected the adeno-associated virus, AAV, because it can't replicate on its own. It has the ability to target very specific tissues and has shown long-term expression or generation of the enzyme. And it's also been shown to be safe in multiple clinical trials and was the type of virus, as Karen mentioned, that was used in the first commercial gene therapy, Glybera. And purposes, we've honed in on specifically a serotype of AAV called 9, serotype 9. We like this one because it has the ability to cross the blood-brain barrier and additionally is able to reach many different types of tissues throughout the body. So it's clear that the diseases that we're working with have issues that appear not just in the brain, but throughout the kids' bodies. And so, as I mentioned, with the use of AAV9 specifically, we're able to deliver by intravenous injection. And that allows the therapy to reach all parts of the body, including the brain. And I want to be sure to state that this particular type of AAV does have the ability to cross the blood-brain barrier. And so what we see and have witnessed in the animal models, that is, those are animals that have the same genetic mutation or mimic the disease or the symptoms, is just that delivery and expression is seen throughout the body as well as the central nervous system. Within about 2 weeks or less, these animals show a clearance of the waste products from the cells. And eventually we do see a return of the neuromuscular and cognitive functions. And that's something that not only great excites us about this data, but it's also equally exciting, if not more, that we see a demonstration of a significant improvement in survival in animal models. So what's next? We look at what is next for the 3 lead programs I mentioned that we recently received FDA approval for our phase 1/2 IND for the Sanfilippo 3A. This is soon going to be starting at Nationwide Children's Hospital. Continuing our work to bring the MPS 3B program to clinic as well. And the Juvenile Batten program, we've completed some early discussions with the FDA that will enable us to begin toxicology studies. And that's a necessary important step before we move into having a clinical trial, human clinical trial. Again, it's really important, worth rather that we partnered with some amazing researchers backed by some solid institutions. Our programs have data, and we look forward to translating that clinically. The slide I mentioned earlier, I'm just under a year into this role. I feel very fortunate to have met with so many of the communities within the communities that we serve. This company was founded, as I mentioned, with families and foundations who believe in the potential of gene therapy, and I'm backed by a leadership team that continues to value this important relationship. And a wonderful example of how community spread throughout the world can really come together to support the science, work industry, and as we've started to bring a therapy to human clinical trial and hopefully well beyond.
I asked Karen about the obstacles to getting gene therapy to work, but I'm wondering what you've seen from the developer side and whether any of these obstacles help inform what might be the best early diseases to target.
Sure. Karen shared about some of the challenges that gene therapy has faced, but it's available with the new vectors, understanding more about just that delivery, getting the vectors to the right place for the right diseases. Abeona's decisions have been very data-driven. We've not only a demonstration of safety, but as well improvement and benefit in animals modeling the diseases. Process, Karen touched on this as well. Manufacturing process for these diseases, it's not simple, it's not always straightforward, and the regulatory steps can also be somewhat challenging. All things considered, we and others in the space are continuing to work through it.
What problems with enzyme replacement therapy that's been used for MPS diseases is that it doesn't penetrate the blood-brain barrier. There have been great strides in treating MPS diseases, but over time they can still take a neurological toll on patients. You alluded to this, but in the case of these diseases with gene therapy?
And, going back to delivery, we're approaching this gene therapy in a manner that addresses not just, the central nervous system and the issues that are happening there, but all throughout the body. I think it is one of those things that time will tell as far as just how long those impacts can last. And that's a question that I think all are being faced with in the gene therapy field.
You've had specific conversations about clinical trials with regulators. What are they concerned about, and how do they view endpoints in trials? Are they different than what might otherwise be expected for the same diseases?
Well, sure. So in general, regulators, I think, are still very focused primarily on seeing a new therapy ushered in a safe manner. So that's regardless of whether it's gene therapy or an enzyme replacement therapy or otherwise. Just their advocacy efforts, I think that there's definitely a great understanding by the agency of how this population is impacted by rare diseases, especially over time. And it's a very different set of risk considerations, or even just a viewpoint of what a benefit might be for someone, take a family living with Sanfilippo, for example, versus another family who maybe is impacted by a chronic heart condition. So, I think the agency is definitely working to understand, and although it's probably not moving at the pace that everybody would love it to, they are making changes that I think will realize as far as benefit in the future. And as far as endpoints, something that is still a significant consideration, again, be it gene therapy, be it enzyme replacement therapy, or another mode of delivering drugs.
Well, the person in your company charged with engaging with and speaking for patients, what's that interaction like for you? Do you find patients have realistic expectations for gene therapy and understand that there are risks involved?
That's interesting. I've been very fortunate to not only be able to interact with the families, but the kids themselves, person, which is my favorite way. I would say that in communities that I've been interacting with and working closely with, that there's definitely a spectrum of understanding on gene therapy and what the possibilities might be. That's all the way from someone who's just brand new, doesn't know, is all the way to those who are actually very savvy and are at the caliber of reading and understanding scientific journals and able to really engage in a scientific discussion, and everybody else in between. I work hard to try to meet people where they are and bring them the information that can help elevate their understanding. And as far as what people are viewing gene therapy as, again, I would say that too is a spectrum. That there is the extreme of, it's the end all be all, the panacea, if you will, all the way back to the other extreme where it's a bit frightening. It's something that's very new, and that's why this particular webinar is especially wonderful and helpful, because it's bringing about this understanding or a base education for all on what GIP is.
Remind our audience that they can instant message questions they have for our panelists as we go, and we'll try to work them into our conversation as best we can, or save them up for the end. If you do have a question, be sure to send it to the All Panelists button. That way I will get to see them. Let's turn now to Paul Fraze, a career NFL lineman. Paul and his wife, Allison, founded the Joshua Fraze Foundation 20 years ago in the hope of finding a cure for their late son, Joshua, who was born with rare and deadly neuromuscular disorder, tubular myopathy. They have continued to forge a path towards clinical trials for his peers, having raised more than $6 million, which has initiated a research program focused on MTM and other related congenital myopathies. Paul?
Daniel, thank you so much. And first and foremost, Allison is very sorry that she could not attend the webinar. She is doing favorite pastimes, though. She's on a patient advocacy mission of sorts, and she's about 90 minutes outside of Rome with 3 families from Italy and their 3 MTM children. So she's just enjoying a wonderful time of visiting and with people that she's been speaking with via the internet and telephone for a number, for a few years. So obviously we wanted to, Allison wanted to introduce you to Joshua, this first slide here, and really she wanted to stress that a child is what pushes parents to be involved at the level that we've been involved with, and Joshua was the reason, was our only reason involved in this space. The picture to the right was taken just 20 days before Joshua passed. I know there were a few questions that were presented. Daniel, did you want to do it in a Q&A type of format, or would you just like to?
Why don't you just give a little overview here, Paul, if you don't mind, about the work you've done?
Okay, great. Well, so Josh was born in '95, and we found out within about 5 months that he was 1 in 55 known cases of myotubular myopathy across the planet, across the globe. So we decided, Allison is very driven. I always say it's her New York chutzpah, growing up in New York with four older brothers. But we really felt it was necessary, knowing 155 known cases worldwide. There's no money going to research. There was only 2 or 3 medical articles that had ever been printed, and a couple of them were from 1967. So we decided to, Allison told me that we were going to use my NFL platform to push hard to raise a bunch of money and raise a bunch of awareness. And, we never looked back. We met with some wonderful scientists that all of them were altruistic to the point of advancing science to get it to the patient rather than having, personal objectives in the research. And, Allison was the driving force behind the fundraising. She just got intricately involved with all of the researchers. She spoke with our scientists on a weekly basis, if not, there were many times on a daily basis, and been able to push forward. I'm leaving the details, and I hope we'll get back to them, through some of the questions. But, we came to the point where we were having proof of concept, and we had to make a decision on what type of therapy we were really going to push and get behind. And there's been a perfect storm, as you all have seen here from the presentations, that what has been taking place in the space of gene replacement therapy. Obviously, the devastating blows in '99 and then 2002. But everything in our situation really has come together in a perfect storm. Now, the slide you see there, we call them our rock star team. Of course, Matt Patterson on the left is the current CEO and founder of Audentes Therapeutics, and he has been it's just been amazing. Obviously, we knew that your million we raised was just a drop in the bucket in order to get from the bench to the bedside. And Alan Beggs is the next there. We've been involved with Alan. We were introduced to Alan Beggs at Boston Children's Hospital and Harvard Medical Center through Anthony Atala, Dr. Anthony Atala, who was a star in the regenerative medicine. And we reached out to Anthony, and he set up a team for us. And then Alan, he's just a master collaborator, and he hooked up with Anna Boujbella, who was over in France, and Anna had been doing some research and actually created a knockout mouse. And then when we were ready to commence canine trials, you'll see Casey Martin Childers there on the far right. We perfect storm, because had we met, we've been ready for Casey if we'd met him 6 months before. But we were ready. Yes.
Can I just interrupt here? You touched on this, but one of the things Allison did was actually help researchers identify and access a large animal model for MTM. How did that happen, and what did she do?
Well, it's quite a story. In his collaborative nature, had been speaking with veterinarians. We all knew we had proof of concept in mouse model, and we all knew that the FDA was not going to allow it to go from mice to human. So Alan had started working in collaboration with some veterinarians. He introduced Allison to a particular vet that was felt like they were looking at MTM muscle tissue under the microscope. Allison, through working with the first veterinarian, was able to get to the origination of that tissue. It was from a veterinarian up in Canada, up in Saskatoon, Canada. Well, we lost the first dog due to some complications and quite frankly, in spam folders and institutional issues. So we were just devastated. And then Allison went to working with the Canadian veterinarian, and within just a few weeks, they had made some calls, random calls to owners that they felt had the dogs from this particular genealogy. A boy by the name of Vic Ketchman calls back. Allison gets on the phone with him. In 5 minutes, Vic says, "Allison, I want to give you my dog. I want to help your son." So because we had lost the first dog, I had told Allison, "You're getting on a plane the next time we find one, if we're able to find one." She was on a plane within a week after that first conversation with Vic, and brought that nibs back to Wake Forest Institute of Regenerative Medicine, where Dr. Childers had been on Anthony Atala's team. Anyway, within the perfect storm, within just a few weeks, nibs went into heat, and 57 days later, we're looking at 12 puppies, and there's nibs, a picture of Allison with nibs in Canada. And then, obviously, to the right, we were able to go up and visit with those 12 puppies. And that one that Allison is holding, of the 12 puppies, 5 of the 8 females were terriers. 1 of the 4 males was affected. And that is a little gray guy right there that she's holding. So, it, obviously, to genetic probability, it was perfect. 25% of the males was. So
how having a large animal model for the disease changed the prospects for developing a gene therapy?
Well, I mean, obviously, again, the mice were responding. We had powerfully robust data from the mouse colony. And, Anna had proven that, and Alan was also proving that with our mice in Boston. It's, as we all know, or maybe not so well, the FDA is not, there's a lot of things that work in mice that do not work, or have not proven to work in humans. So obviously want to have a larger experimental model that is more akin to the human system, and the canine model is a very good system and recognized by the FDA. So the bottom line is, if we were able to have proof of concept as robust as from the mouse colony to the canine colony, we felt very strongly that the FDA would consider our data and allow for clinical trials.
So, Untitled Therapeutics, you mentioned, is developing and commercializing the gene therapy. What did your foundation have in getting a company to pursue this, and how is your interaction with the profit company different than, say, with academic researchers?
The initial contact actually came, interestingly, we were having a kind of a Lorenzo's Oil-ish dinner with researchers, Martin Childers and Barry Burns from the University of Florida, and it was the first time Barry had been exposed to the MTM research, patient advocate, and I think Casey had kind of stalked him at one of his presentations a few months before, and invited him to at least join in our team, because his involvement with gene replacement therapy, therapeutics. So anyway, Barry, by the end of the dinner, Barry said, "You need to meet this gentleman, Matt Patterson. He's with OrbitMed." And Barry made an introduction. That the introduction couldn't happen fast enough. Casey, Martin Childers, was in the process of moving from one institution out to the University of Washington. So he was delayed in being able to talk to Matt Patterson. And I said, "Well, Matt, if you wouldn't mind hearing it from a father or a mother, I'd love to chat with you." And we had a wonderful conversation the first time. Matt has an altruistic goal of helping people as well. Obviously, there's a money-making venture in place, and that's a big part of the whole system as well. But I was able to present to Matt. Interestingly, he said, "You sound like you guys are doing a good job. I'm going to watch from a distance, because gene replacement therapy is still a little bit off bounds for VC, but he continued to talk to our scientists, and that was literally just a few weeks before Glybera got approved, and that really obviously validated our stance and Matt's ability to put together a presentation, which quite frankly he was able to, over a period, raise about $130 million. So how has that affected how we've been involved with the science, or Allison's been involved? Again, Allison used to speak with our scientists sometimes on a daily basis. We knew everything. I was in the laboratory to witness two of our just rock star dogs who have actually become studs. They were affected, treated, and now running around like normal dogs. The histology has normalized, and they are used as stud dogs to increase the probability of the canine model. But obviously, for many different reasons, Allison was able to work very closely with Matt early on and get him in touch with other patient advocacy groups and other scientists. But we don't know exactly what's going on behind closed doors, although we get some wonderful updates periodically. And you're mentioning the manufacturing and the regulatory issues are very complex. Matt said this is the most complex, since he's ever been involved with, regarding genetics, and so it's in good hands. We feel very strongly about that, and Allison continues to work with dentists and other patient advocate groups.
A few minutes left to take questions from the audience. If you do have a question, please send it through the instant messenger at the bottom right-hand corner of your screen. If we don't get to your question, we'll follow up and try to get you an answer via email. We've had a question about any updates regarding spinal muscular atrophy and the AAV9 gene therapy program. Michelle, is that directed to you?
It quite possibly could be. I mean, that is, again, utilizing the same AAV type that Abeona is utilizing for the 3 lead programs. I do know that they have begun and have been treating, I believe, over 10 patients. And I don't think that they've released any data on that just yet, but we're all looking forward to seeing that. Just as a side comment, in addition to the Avexis trial, has been demonstrated to be safe in other clinical settings. So this is also technology that was derived from Nationwide Children's Hospital. So quite possibly directed at me.
And I don't know if any of our panelists are able to answer this one. We have a question about how does the vector for Hunter syndrome look?
Karen Kasarski, there are a few things in development for Hunter syndrome, which is also known as MPS II. And there is certainly one under development by NextBio and by another company that are both using AAV9 vectors, the same vector that Abeona is using. And there's also some work under development from a group in Italy trying to utilize a different form of gene transfer using a patient's own stem cells. And that one's trickier in some senses in that it may only be useful in pre-symptomatic patients. I think time will tell. So there's a couple of different approaches looking to get to the clinic in the next couple of years.
A lot about new gene editing technology, CRISPR. This has enormous ramifications in the area of research. If anyone can speak to its potential impact on gene therapy. Karen, weigh in on this?
Sure. So it's an area that certainly intersects well with gene therapy in terms of trying to deliver the machinery you need for this type of editing. So the goal behind CRISPR very often is to actually correct a mutation within the genome, so within the chromosomes of the cells. So rather than adding a gene back, on top of the mutation, essentially, it would be looking to try to correct what's there. The technology can also be used in various other ways to work around issues in genetic defects. And, you know, it's a really fascinating technology, a very rapidly evolving field, and there are several companies looking to get into clinical trials. They're hoping over the next few years. I think if you're using it to try and treat a specific genetic disease where you're trying to correct the mutation, it definitely would be useful for only a subset of patients because different patients have different mutations. So in diseases where there are certain common mutations that are often repeated across, you know, with a high percentage of that patient group, it may be more useful there perhaps. But it has a lot of promise as an additional technology to be used in gene therapy.
Pricing is an area of growing controversy. Gene therapy is expected to be expensive, but it's also a potentially curative therapy. I'm wondering if any of our panelists have thoughts on how payers view this. Is it like we're going to see new payment models emerge from this to address gene therapy?
Well, a run at this. I mean, it's a significantly topic, and you know, we've seen already, you know, over in Europe with the Gleiber's pricing. You know, that's kind of sent a shock throughout. I guess in the world with regard to that particular price tag. What I'd remind or what I bring people back to is that, you know, what's currently available now by way of an enzyme replacement therapy strategy where, you know, that might be administered every other week or something to that effect, that's something that a person is on for life. And a price tag currently of about maybe $200,000 to $400,000 annually, you know, that is definitely something to compare to or to consider as we look at how to untangle or how to solve reimbursement for gene therapies. The other thing I would add, too, is, as Karen pointed out in her presentation, the approach for gene therapies, it can be very different. There's no one single method, and so I think that, too, adds to the complication of figuring out reimbursement strategies. I know that's not necessarily exactly helpful in providing an answer, but these are the considerations that everybody involved is going to be looking at.
Before we go, I want to ask each of our panelists the final question. Do you believe gene therapy will become the standard way we treat genetic diseases? If, why not? And if so, how long will it take? Karen, why don't you begin?
So you've asked a very broad question, but I think it will become a standard way to treat many genetic diseases. It's with current technologies and current knowledge, we have to start with diseases where we know where the genetic defect is, and it needs to be a disease for which we have the right types of delivery vectors in order to be able to treat that particular disease. So I think it has the potential to become the standard of therapy for a certain subset of diseases. You know, I hope for a fair number of them, and that there are other diseases around in which I think we need to find newer delivery systems that will work for those. And these things are evolving more and more. Items keep getting added into various companies' pipelines. So I see this as really developing and taking off in the next 10 years.
You.
Well, you know, just to kind of recap what Karen shared, the potential is most certainly there. We are seeing things that are very encouraging and exciting in animals, and that's now starting to be witnessed in human clinical trial. We're also, you know, cutting with these results are strides being made with regard to manufacturing, scale up, as you heard. Those are some challenges as well, but the solutions are converging basically with the being made in clinical trial. And, you know, we look ahead, the vectors and the other tools that are being created are truly, you know, working to catch up with the imagination or with just the creativity of the science itself. So I would agree within the next 10 years, you know, things could look significantly different. But I would even say that even sooner than that, as with what's mentioned here, Spark, other companies, Bluebird, Nightstar, we will see some amazing results and build upon that.
We have a patient advocate's perspective on that?
Oh, absolutely. We've been very fortunate, and I love how Global Gene simplifies a lot of things about rare disease. And one, there's 7,000 plus rare diseases out there. We know that there's a handful that are similar in structure to MTM, to Joshua's disorder. His is a small enzymatic protein, and one of the reasons right now that gene replacement therapy is working so well, I mean, it's working well with very small proteins. That's what really benefited us as an MTM1 genetic disorder. Now, we think there's a handful, if not maybe hundreds, of disorders with small protein issues. And Begg simplifies it for me. He says, Paul, MTM is like replacing the windows, but there are other disorders with very large proteins that is like replacing the inner girders and the supporting structures. So we're in a perfect storm. I do believe science has advanced exponentially in the 20 years we've been involved with it, and things are just happening more and more quickly. And we're excited about the prospects of gene replacement therapy. Regarding MTM, we know that it may work. It's a monogenic situation. It may work on some, most, maybe all. But the probability is there will need to be other supplemental therapies out there. So we continue to look for new therapies to help support what we've done so far.
Well, I thank our panelists for sharing their insights today. Our panelists are Karen Kozarski, President of Vector Bio Partners, Michelle Berg, Vice President of Patient Advocacy for Abeona Therapeutics, and Paul Fraze, founder of the Joshua Fraze Foundation. Thank you all for joining us today. I'd also like to thank the staff of Global Genes, and in particular Ashley Yee, for her great work putting together and coordinating today's program. Here to join us for our next webinar, Strategies for Effective Fundraising, which will be held on June 21st. A reminder that the Global Genes Toolkit on Gene Therapy will be available at the end of this month. Also, registration for Global Genes Summit will be opening in May, so keep a lookout for that. I hope you'll join me every week for our Rarecast podcast, which you can find on the Global Genes Rare Deli or on SoundCloud, iTunes, or Stitcher. And you can continue the conversation with us by following us on Twitter. You can find me at DS Levine and Global Genes at Global Genes. As you seek to learn more about rare disease and connect with others, I encourage you to explore the many resources available on the Global Genes website. On behalf of Global Genes, I'm Daniel Levine. Thank you all for joining us and being part of today's webinar.